Okay, before we get talking about how muscle contraction works and tie that all into depolarization and repolarization, the whole basic premise of this that you've got to remember is that the inside of a cell, a normal cell at rest, inside is going to have a slightly negative charge. Outside, it's slightly positive. So you have this, what we call electrochemical gradient. You've got this charge difference. It's not neutral. It's negative inside the cell. And why is that? You also need to be familiar with this as to why. Inside the cell, potassium typically is higher concentration inside the cell than outside the cell. Sodium and calcium are typically very low concentration in the cell, but high outside the cell. All of these have a plus charge to them. Calcium actually has a +2 charge to them. So if you have more of a plus charge outside the cell, that's going to give you a slightly negative or slightly positive charge outside, slightly negative inside. You need to remember which one is higher where, because in a moment, as we go through the steps of muscle contraction, this will explain why things move in the direction they do. Remember with diffusion, things want to go from a higher concentration to a lower concentration. So oftentimes what's going to happen is we have these ion gates, these gates that will open up in the plasma membrane. Some of them are very specific for which ion can pass through. So let's just say we open up a calcium ion gate. If we open a gate and calcium's higher outside the cell than inside, by diffusion, it's going to want to flow in. Same thing for sodium. Sodium is going to want to flow into the cell, but potassium is going to want to flow out. So at rest, we have this imbalance of the concentration, and that sets up for the charges, which is the resting potential, that you've got this electrical charge. Why? Because you have a difference in the charges on either side of the membrane. Why do you have that charge? It's because you have different concentration of these ions. And we're going to be making adjustments of that in order for an electrical impulse to be generated, come down and stimulate the muscle for muscle contraction. So you've got to keep this in mind and refer back to it. Okay, so we're going to go through the steps of muscle contraction in detail. I've got it down to about 27 different steps, so it's a lot of detail, but if you just take it step by step, it makes sense. This is the axon up here of the neuron, so the main cell body would be further up. I'm not drawing that in. But the electrical impulse has been generated. There's a stimulus, stimulated the neuron, and so you have your action potential that is moving down your axon. So this electrical charge is moving down towards the axon terminus. That's the little knob on the end of the axon. Remember from before that the whole premise of this is the inside of the cell at rest is slightly negative. The outside is positive. When the axon action potential is coming down the axon, it's changing. So temporarily the inside is positive and the outside is negative. So here it's still negative inside, positive outside. As that action potential, if I can speak, moves down, this is changing. So all these, you're getting this change in the charge. As it hits this knob on the end that's called the axon terminus, what happens is you have some gates, some ion gates that are voltage gates. So this is a voltage, this current that is moving down, this action potential. When it hits that gate, that change in that charge opens the gate. So it's like someone just came in and opened the latch on a gate, or just opened the door. Well, if you open the door, and these gates are specific for ions, in this case it's specific for calcium. So if you start opening the gates for calcium, calcium is higher outside the cell than inside the cell. So diffusion says, where does the calcium go? Calcium is going to flow into the axon. what does the calcium do once it's inside? In the knob, you have these little vesicles. They are filled with the neurotransmitter acetylcholine. Now, in this case, we are looking at a neuromuscular junction, so we're looking at the connection of a neuron to a muscle. This will be a muscle over here. With skeletal muscle, the neurotransmitter is always going to be acetylcholine. Now, if it's a neuron to a neuron, there's a possibility that that neurotransmitter could be a different chemical. But in this case, it's acetylcholine, which is what the ACH abbreviation is for. So what's a neurotransmitter? It is a chemical that is released from a neuron, and it's going to have an effect on another cell. So how does it get released? Once again, calcium influxes into the axon terminus. It flows in because of this action potential that was moving down. It is going to trigger these vesicles to move towards the plasma membrane. And what's going to happen is as it moves down here, it is going to spill out all of those chemicals. All of that acetylcholine now is going to flow out into the space here. And what is this space right here? The space between the neuron and the muscle. If I can write just here, draw this arrow. That is your synaptic cleft. Remember, the neuron and the muscle are not physically touching each other. There's a little tiny little bit of space between the two. That is the synaptic cleft. So when the acetylcholine is released from these vesicles, why? Because the calcium triggered it. How did the calcium get in? Because the action potential came down and opened the gates, allowing calcium in. So you release the acetylcholine, you're releasing it on the neuron side. So by diffusion, that just means it's going to flow from higher concentration here where it's being released across to the muscle side. There are receptors on the muscle side that are very specific. Once again, remember there's a lot of specificity here, which the acetylcholine will bind to. So it will bind to these receptors right here. Remember, whenever you have binding of two things, there's usually going to be a shape change. And so now what happens as it binds, now is the acetylcholine going to bind to this receptor? No, because that's for something else. So it's just going to bypass it. But it binds to the specific receptors. And what that does is it opens another gate. And in comes sodium. Now remember, sodium in a resting cell is normally outside. It's a much higher concentration outside the cell. So when you open that sodium gate, you're going to have sodium flowing which direction? Inside, because it wants to go from high concentration to low concentration. So you now have this sodium, which is a plus charge, flowing inside. Remember, a cell at rest is normally negative. So if you're normally negative and now you have all this positive charge coming in, you're going to start to change the charge inside this muscle now. Through the whole cell, not initially, just in this region where the sodium is coming in. Well, if you have the sodium coming in and coming in, it is going to now trigger some of these other gates, which are what we call voltage gates. So they are opened by a change in the charge. You're changing the charge. So these are now going to open. What are they specific for? Also sodium. So now you thought you had a little bit of sodium coming in before, initially right here when you had the acetylcholine bind. Now you open up the voltage gates. So you have this massive influx of sodium. Once again, what's now happening along the membrane surface of the muscle? This is all becoming positive charge now. You've just generated an action potential in the muscle. You reach a threshold point It's all or nothing. It's kind of like, okay, the cell's slightly negative. You got all this positive charge in, so the charge starting to get positive. You reach a certain threshold and boom, it's gone. It becomes very, very positive inside. That's your action potential when you reach that threshold point. Before that, it's what we call a greater potential. You just had this little area where the changes. Now it's spreading because you've had so much positive charge coming in. That positive charge is going to continue this action potential all the way down the membrane until you reach the T-tibials. Remember the T-tibials, these are these little indentations in the cell. So the charge is going to be coming along that membrane, it hits that T-tibial, and then yes, it's going to move down. What is the T-tibial part of? It's part of three things. Collectively, it's called a triad because there's three things. What are those three things? You have a T-tibial in the middle, and then on either side you have what's known as a terminal cistern. So terminal cisternoid, T-tibial, terminal cistern. That together is a triad. What's the importance of this? Guess what's stored in the terminal cisternoid? That's where the calcium is stored. Calcium normally is high outside the cell. If it's in the cell, it's going to be stored. It will not be free floating in the cytoplasm. In the muscle cell, we call that the sarcoplasm. It's not going to be free floating here. It is going to be kept in storage. Think of a cistern, like a water cistern. You store water there. Well, in the cell, the terminal cistern, you store your calcium there. When that action potential reaches those terminal cisternoid, the triad, that calcium that is stored is now going to be released out into the sarcoplasm. So it's like you open the doors there and you're releasing it all. And it's all coming into the sarcoplasm. And why is this important? Because we need the calcium in order to start the actual muscle contraction. We haven't gotten to contraction yet. You're at the what we call the excitation phase. You're getting the stage set for the contraction. Before we continue further with the muscle contraction, just as a reminder as to how the setup is. The blue line is your actin. That's going to be your thin filament. Remember, there are two sets of proteins that are associated with it. The orange is a protein strand, that's your tropomyosin. Mask so you can hear me better. So the orange is the tropomyosin. The little black dots, that's the other protein, troponin. What happens is calcium is going to come in and it will bind at the troponin site. Whenever two things bind, there's always a shape change. So what happens, calcium binds to troponin, which is attached to the tropomyosin, the orange strand. So when calcium binds to troponin and there's a shape change, that's going to move the tropomyosin, or that's going to move this orange line. And why is that so important? It's because that orange line is covering up sites on the actin where the myosin, or the thick filament, binds to. Without calcium, those sites are covered up, so you don't have any binding. But if you expose that active site on the actin itself, that binding site, if you expose it, then the myosin can bind, and then you can start muscle contraction. So how do you expose it? You have to move the tropomyosin. You have to move this orange line out of the way. So how are you going to move it out of the way? Get the calcium to bind to the troponin. So it's as though if this meter stick were to be your actin, the thin filament. And with my lanyard, you try this. That is the tropomyosin. And so it's wrapped around it. Now, if the calcium comes, and let's say this black clip, hopefully you can see that, that is over an active site. If calcium comes in and it binds And now it shifts everything. Now your active site's exposed, so you can get binding here. You couldn't get binding before because it was covered up. So when calcium is present, it binds to the troponin. That binding causes a shape change, which shifts the tropomyosin, now exposing the site on the actin so you can have binding occurring. So you had your calcium being released from the terminal cisternae over here in that triad portion because the action potential came down. It released the calcium, and then remember we said the calcium is going to play a role with the actual contraction that's going to occur. So down here I've drawn the blue lines are your actin or your thin filaments. The red is your thick filaments or your myosin. So as calcium is flowing into the sarcoplasm, remember that's the cytoplasm of the muscle cell, which is where these myofilaments are, these muscle filaments are. We're all just inside one muscle fiber, one muscle cell. So the calcium comes down, and if you remember, I don't have drawn on here, the actin, from what we just previously said, has the tropomyosin wrapped around it, and on the tropomyosin is the troponin protein. So in a nutshell, calcium comes down, it's going to bind to those proteins, moving them, exposing the binding sites. So now, remember the myosin has those little heads on them, those connecting points that can now bind to the actin. Why didn't it bind before? Because it was covered up. Why is it now uncovered and exposed? Because the calcium moved, was the trigger to move everything. And so this is what we call cross-bridging. That is when the myosin heads, we call these heads because they look like little knobs, they bind to the actin. And so what happens is now, sorry, you're gonna have to use my hands as a demo here. If this arm is the thin filament, and my right arm right here is the thick filament, and it's got the myosin head, the binding site is now exposed so the head can bind to it. It binds to that site, the head cocks, and then it's released. This series of steps will involve ATP. It's going to bind to the next site, cock, release, bind, cock, release, bind, cock, release. This is called the sliding filament theory. See how it's moving the thin filament along, which I couldn't really draw here, but you would have the blue lines moving this way. So it's shortening this way. That's contraction. You are finally getting muscle contraction because you're shortening that muscle now as it's sliding like this, because the binding site is exposed. That will continue as long as you have calcium here. How do you relax the muscle? How does all this stop? Well, you need to stop the action potential. How do you stop the action potential? You need to go way back over here in the synaptic cleft where you had the acetylcholine, that neurotransmitter that was being released that's opening these gates. Shut the gates, close the gates. It's kind of like close the front door. Don't let anything else in. How do you do that? There's an enzyme called acetylcholine esterase that will break down this acetylcholine. And it starts to break it down, removing it from those receptors. When you move it from the receptors, you close the gate. If you close the gate, you are now stopping this influx of sodium. How do you get things back to normal? Because you had a lot of positive charges coming in. Remember, you turned the inside of the cell positive. At rest, it's negative. You need to get it back to that negative charge. So this is where now your sodium gates are going to close. Shut down. Don't let any more sodium in. That's going to help because you're not letting any more positive charge in. You also have some of these other gates which are specific for potassium. They are going to open up and they are allowing potassium to flow out. Potassium is normally, remember, higher inside the cell. So if you open a potassium gate or door, it's going to flow from high to low. It's going to flow out. And it's a positive charge. So now you're starting to move some of that positive charge out. The other thing that's going to play a role, and that you have to remember and keep in mind, is that at all times, during rest and at contraction, your sodium-potassium pumps are always working. And their purpose is to try to keep this imbalance, and that may sound backwards because chemically everything's wanting to reach equilibrium. You're wanting to get the potassium out, return the inside of the cell to more negative charge, but do you want enough to go out so you have equal concentration on both sides? No, you do not. Why? Because if you had equal concentration of potassium and sodium on both sides, inside and outside the cell, you would never have this electrochemical gradient that we talked about at the very beginning. If you have equal concentration of sodium on both inside and outside the cell and you open a sodium gate, are you going to have sodium flooding into the cell? No. It's already at equilibrium, so nothing would flow in. You would never generate an action potential, which would mean you would never get a muscle contraction. You would never get a nerve impulse. Bottom line is you would die. So you have to maintain this difference. You have to, after a muscle contraction, as it starts to relax, you have to get the inside back to a negative charge, restore how everything was. The sodium-potassium pump is going to take 3 sodium ions out. So you're getting 3 plus charges out, and it is going to be bringing in 2 potassiums. Sodium and potassium each have a +1 charge. So if you're taking 3 plus charges out but only bringing 2 plus charges in, then you're helping to keep the outside positive and the inside negative. So that's going to also play a role in helping to restore things to that resting potential. Restoring that inner charge back to that negative charge rating is repolarization. Until everything is back restored to how it was before you even start this whole process, you're not going to be able to get another impulse. You may get an impulse on another cell, but on this particular cell, it cannot respond to another stimulus until everything is back restored to the resting membrane potential. Okay, this chart is my attempt to help kind of put together with you what's going on in terms of the charges in a cell during muscle contraction. And when we talk about depolarization and repolarization, how all that ties in. So at rest, we've talked about how the inside of the cell is slightly negative. And we can actually measure that. It's at about -70 millivolts. So that would be the resting stage. When you have initially that impulse, if we're talking about on the muscle, where the neuron stimulates it, you have the acetylcholine come in, bind to the receptors. Initially you have the sodium gates start to open. So you have those sodiums coming in, which is a plus charge, which means you're going closer now to 0 for that charge. You're going more positive. Why? Because your sodium gates are open. And initially you have them open, and then all of a sudden the voltage gates open where you have that huge influx of sodium and you get this dramatic rise. Threshold is about -55 millivolts. That's the point where if you can reach that point, you got action potential. If you don't quite reach it, nothing's going to happen. So that's sort of that magic number of all or nothing. You got to get to this point. Going to a more positive charge, so this yellow line here is 0 millivolts. You're going to go up to about +30 typically. As I said, during this rise up on this peak here, your sodium gates are open, so you have all that sodium coming into the cell, making it more positive. That is depolarization. When your sodium gates close, So now no more sodium's coming in, and your potassium gate's open, and remember potassium is moving out of the cell, so you're taking that positive charge out. Now you're starting, because you're removing positive charge, you're starting to become more negative, and this is what we call repolarization. And that's going to continue on down, and sometimes you kind of overshoot a little bit. If you overshoot, technically this little area where you've gone even more negative than what the resting potential is, that's called hyperpolarization. But it will very quickly come back to the resting potential, the resting state right here. So depolarization, you're becoming more positive. Repolarization, you're returning, becoming more negative again, trying to get back to how you were at rest. While you are in this stage here, until you return back to rest, you cannot respond to any additional stimuli. But hopefully that just kind of helps you to see it put into a graph in that way.